Hello everyone, welcome to my poster. I would like to introduce you our recent progress on a nucleosome calling package SEM, which is able to characterize nucleosome positions and nucleosome types from amnesic data genome-wide. To start, as we know, canonical nucleosome is a histone octamer wrapped by DNA fragments. In recent years, there have been evidence supporting the existence of diverse forms of nucleosome structures on the genome, including hexamer, prenucleosome, half-nucleosome, and tetrasome. In addition, there have also been evidence supporting the existence of a type of amnesic-sensitive nucleosome, which is called fragile nucleosome. Fragile nucleosome attracts researchers' attention because it's very close to the transcription start site. It has been shown that the various subnucleosome structures are associated with gene regulation and DNA replication. However, we still lack an effective bioinformatics method to distinguish various nucleosome types from amnesic data. To resolve this issue, we developed the size-based expectation maxima algorithm, in short SEM, that can distinguish nucleosome types by protected amnesic fragment lengths. In brief, SEM first runs a Gaussian mixture model on overall amnesic fragment length distribution. Its distribution corresponds to a nucleosome. For example, a canonical nucleosome should protect 150 base pair DNA fragments, which fits the blue distribution here. After getting the nucleosome type length distribution, SEM computes the probability of each nucleosome generating each amnesic fragment, considering both the location and the length of the fragments. After the expectation maximal, we get the proper assignments of each fragment to each nucleosome, and we compute the mixture probability of each nucleosome based on the assigned fragment length. After developing SEM's package, we compare SEM to other existing nucleosome calling packages on common nucleosome matrix, including nucleosome diallocation, nucleosome occupancy, and nucleosome fuzziness. Overall, the results suggest that the SEM has comparable performance to other nucleosome calling packages on these matrices. The most important feature of SEM is to distinguish various nucleosome types from a single amnesic experiment. To demonstrate this feature, we apply SEM to a low-dose amnesic chip-seq IPH2B dataset in mouse embryonic stem cell. SEM discovers three nucleosome types, including short-fragment nucleosome, canonical nucleosome, and dinucleosome. Short-fragment nucleosome is highly enriched at the transcription start site, which is consistent with the properties of fragile nucleosome previously reported in other studies. Further, we want to compare the short-fragment nucleosome to the previous reported fragile nucleosome in terms of amnesic sensitivity. We accomplished it by taking the log ratio of the number of amnesic fragments under low amnesic digestion versus high amnesic digestion as a proxy of amnesic sensitivity. Surprisingly, short-fragment nucleosome in open chromatin show a very high amnesic sensitivity as fragile nucleosome. So we define these subtype of nucleosomes as fragile nucleosome in our following analysis. We further explore the relationship between the fragile nucleosome and the various factors genome-wide, and found that several chromatin remodelers, including CH6, CHD8, and EP400, are associated with fragile nucleosome. Overlapping fragile nucleosome with candidate cis-regulatory elements tells that fragile nucleosome mostly exist in promoter and enhancers, which suggests fragile nucleosome could be an important participator of gene regulation. In summary, we developed SEM, a new nucleosome calling package that can precisely predict nucleosome diallocation, occupancy, and fuzziness. SEM distinguishes short-fragment nucleosome in mouse embryonic stem cell on a low-dose amnesic chip-seq H2B dataset, and it resembled previous reported fragile nucleosome in open chromatin. SEM defined fragile nucleosome associated with chromatin remodelers genome-wide. That's all my presentation. I hope SEM can contribute to future studies on various nucleosome types genome-wide and help us understand the function of these nucleosome types in gene regulation. I hope you enjoyed the presentation and feel free to ask questions on the website. I appreciate your attention.